My name is Reihaneh Mostolizadeh from Tübingen University. Welcome to my talk. My talk is about a workflow for community model with focus on the human nose and trying the workflow on our example community with species Staphylococcus aureus and Dolosigranulum. The first question always is that what motivates us for such a research and workflow? Of course, there are a lot of reasons which I try to summarize some of them here as you can see. For example, no systematic study on human nose. The research on the human nose is always limited to few species, limitation in cultural capability of strains in the lab, and maybe the more important one, the spreading of infection through the human nose causing tens of thousands deaths per year. Maybe now is the time for doing this research before the next pandemic happen around the world. And of course, this research gives some research question to us and also some promising potential to test for. Let's start with our workflow. The workflow is comprised of several steps like single model, which focus on analysis of all single models that are coming to the community, media, which include different definition of media for the community, objective function, approach that are included, compartmentalize and pool, and the last part, result. In a moment, I go to each step and explain one by one. The first step, like any workflow, is based on the single model. Our workflow does not reconstruct genome scale model. It needs to be available. Our workflow only checks the quality of the model. However, if the model comes directly from our group, checking the quality part can be neglected, since we focus on the reconstruction with high quality and standardization. Then we check the growth of the model on the human nose. In 2014, the Charisma from Tübingen University published an article about synthetic nasal medium which mimic human nose. We use this media to mimic human nose for our analysis. The growth will be checked. If the model has no growth, we check for additional required metabolite to allow the model grow on the human nose. And the last part, FVA computed, Jacquard distance calculated to see the similarity between species from the reaction, exchange reaction, and metabolite perspective. The next step is media. Of course, as I mentioned before, our main focus is the SNM. However, SNM will be also used as a media for our community, but after each model has grown SNM, we create some further media for the community by combining FVA computed for each single model and some further constraint. Depend on the Jacquard distance and similarity between a species, the difference between those media are sometimes 2, 3, 4, or sometimes less or more metabolite. Therefore, the reason for this variation is that to see the effect of each single metabolite in the community. When computational work want to be integrated with experimental work, most of the time the community media are defined as simple as possible to be feasible for testing in the lab. However, this reduction might drive us to the different direction or even wrong result. Therefore, here we focus on the different media. We try to watch the effect of each single metabolite on the community, but on the other side, keeping the feasibility of testing the result experimentally for future validation. The third step is the defining objective function. We use a linear combination, which is extendable if we have more species in the community. The other objective function to give different weight to the growth of each single species in case if one species has faster growth, higher value. Experimentally, this can be also done as people in the lab try to give a head start to one species. Another objective function can be defining a certain value for the community to be achieved and see the contribution of each single species by convex combination for producing of the whole growth for the community, and of course the role of each single metabolite in the contribution. And the last one is the combination of the convex and weighted objective function. The approach we are using for creation of the community is based on compartmentalize and pool. In the compartmentalize, for shared metabolite, we define integrated community, and shuttle reaction. Integrated community, we try to define all non-exchangeable reaction and metabolite with its name and model ID. Then this shows which metabolite belong to which model. However, all exchangeable reaction just be added to the shared environment as they are, then they can be used by both. Compartmentalized can be also defined by shuttle reaction. More difficult to implement it because we have to define which media we are using, which media we would like to be shared, but more reliable for further analysis, and we can see the result with more details, how the change of each metabolite affect on the growth of each single species in the community and in community growth. We can also define the shuttle reaction as we wish. For instance, I would like just oxygen to be shared or glucose or whatever. Put also in our workflow we used, but we assume we have one bucket and put all reaction and metabolite in there. It is so easy to implement it, and especially it is good for like a first impression about the interaction between species. But no details or further analysis is available by this approach. And the last part, like any workflow, is a result. We get different type of result when we are using this workflow. One part belongs to each single model, and we can see how this single model can grow on SNM, what extra metabolite they need, what is their dependency to the different metabolite, and so on. And the second part is that how they behave when they are in the community. What is their dependency on that shared metabolite? How they interact with each other if the common resource is minimum amount or maximum amount. Do they fight for common uptake, or due to different dependency they allow each other reach maximum growth? And is there any metabolite which produced by one and consumed by the other one? Does this metabolite play the role of friendship between those species? And of course, one interesting thing we can also do in our workflow, if one species does not have grow on SNM, if we put it on the community with the other species which they have growth, these species can take whatever it needs from the other species or not. Okay, just our example community is one community between Staphylococcus aureus and Dolosigranulum. We implemented our workflow on this community. The first genome scale model of Dolosigranulum came up from our group in 2021 with my colleague. She predicted Dolosigranulum need these 3 additional metabolites to grow on SNM, and it would grow with the value of 0.28. And the Staphylococcus also we took the model from 2019 with a little bit change, and the model has grown SNM equal to 2.55. One of the results which we get by our workflow. We try to put these 2 models in the community. However, we try to delete it all metabolite which help or require metabolite and necessary for the growth of Dolosigranulum, and then put it this Dolosigranulum without growth and Staphylococcus with growth in one community. The media which we used here, of course, it is the original SNM, because the Dolosigranulum has no growth. Then we cannot do further computation based on FVA and FVA. Then 2 amino acids necessary for the growth of the Dolosigranulum produced by Staphylococcus, but we had to add the other metabolite manually to the community. In general, the other metabolite is also released in the environment bacterial cell lysis, thus becoming available for uptake by DP. But it is a limitation of modeling which we cannot see the physiological feature by modeling. Then we add the other one manually. Therefore, we notice in this community, DP also grow because it takes whatever it needs from the SA. And also some further analysis we did, and then we came to the discussion part as you see here. We also noticed the serine released by DP. Sustain is also available in human nose, which also will be taken up by DP. Lanthionine is formed by cysteine and serine. It was also examined the Dolosigranulum strain exhibiting the strongest inhibition of the Staphylococcus are the only strain that contained both a lantipeptide biosynthesis gene cluster and a bacteriocin. The name of lantipeptide was introduced for all lanthionine containing peptide with antibiotic. Lanthionine have also been found in a bacterial cell wall and are the component of the group of gene-encoded peptide antibiotic called lantiobic, and which is include nisin. Nisin is a family of lantibiotic that bind to the lipid to cell wall of the target bacteria and disrupt the cell wall production. On the other side, Dolosigranulum is included in lactic acid bacteria. Lantibiotic component resulted in antibacterial of the Staphylococcus. This all together brought out to this question: does the release rate by Dolosigranulum, which form lanthiones, including nisin, play a role in inhibiting of Staphylococcus? This remains to be experimentally explored in the future work. This is such a result we can get by our workflow. Thank you for your attention, and also thanks to all people in our group.